Hello. There are many steps and parts to DNA replication, and transcription and translation are some of them. Within each, there are multiple steps that work together in order to move the process along. But I want to focus more on transcription. Transcription of mRNA encoding genes is a multi-step process divided into initiation, elongation, and termination. I'm going to talk about elongation inhibitions, but first, some background. Here is an image of DNA replication just to show all the multiple steps that go into the process. Also, here is a look at both transcription and translation. Transcription is the one I'm going to be focused on more so, which is basically just a rewriting of the genetic code, but keeping it in the same language, for an analogy. Regulation of initiation plays a major role in gene expression, but it has become more apparent throughout the years that transcription elongation also plays a major role in the gene expression. Studies of transcription elongation have revealed that there are existence of several families of elongation factors governing the activity of polymerase II, either positively or negatively. Elongation is the addition of nucleotides to the new DNA strand after the addition of a primer. So here is a little image of elongation. And that's going to have that RNA polymerase moving along the DNA template strand. So synthesis of the growing strand involves adding new nucleotides down here, as we can see, one by one in the exact order specified by the original strand. So like I said, it's transcription, so it's not changing it into like a different language. As I said, it's keeping it the same, it's just almost rewriting it. So during elongation, RNA polymerase walks or moves along one strand of DNA known as the template strand, which runs in the 3' to 5' direction. There is a template strand, and then there's a lagging strand. The template one is the one here running the 3 to 5, and then non-template or the lagging strand is the one up top up here running 5 to 3. So we're focusing on the 3 to 5 or the template strand. This is a very important part of DNA replication, but there are occurrences where it can be inhibited. There's a bacterial toxin that inhibits DNA replication through the direct interaction with the beta clamp. Here's an image, and we could see up here is the beta sliding clamp. The function of the beta sliding clamp is to bind to DNA polymerase III, the E. coli replicase, and then it slides behind the polymerase, holding it to the DNA for incorporation of thousands of nucleotides, but it does this all in one binding event. There's a toxin-antitoxin, also known as TA systems, that are heavily prevalent on bacterial chromosomes. In relation to this topic, the TA SOC-A, SOC-AB is atypical, and it is unstable. It constantly is degrading by the protease CLPXP, and then this degradation requires the antitoxin. SOC-A is the antitoxin that it requires, and it is a protease adapter. And then it was found that the other antitoxin, SOC-B, blocks replication elongation through an interaction with that sliding clamp. and it drives the replication fork to collapse. Here is a look of the replication fork right here where it's circled, and this is where the replication fork will open up for those templates to be read and coded, and then it will close behind as it moves down the stream. So it'll continue to move, it'll open, move down, and then it'll close behind it as it moves. It was also found that SOC-B disrupts replication by outcompeting for the clamp binding, by outcompeting other clamp binding proteins. The toxins characterized by the TA systems inhibit growth by targeting relatively limited set of crucial cellular functions such as translation DNA, replication, or cell wall growth. For instance, some toxins block translation by cleaving the mRNAs or cutting them off, and other inhibit replication by binding to GNA gyrase, which is required to relieve supercoiling, which occurs ahead of the replication fork. Elongation is a very important part of transcription, which is a very important part of DNA replication. So if it is not working smoothly, there are many problems that can happen, especially when we need to create new cells.